This conference will now be recorded.
Hello all, welcome to another video dedicated to Grand Canyon University. Today we will be discussing the role of lung surfactants and looking at a promising study that has nothing to do with the lungs. I will go over basic lung anatomy, but students should have some preliminary knowledge and should know the definition of diffusion. This video is for higher education students with a decent background in anatomy, biology, and chemistry. Let's get into it. We'll start by briefly reviewing the anatomy of the lungs, hitting on the major areas and functions. The diffusion of oxygen and carbon dioxide are obviously the most important functions of the lungs, so we will review how those gases are exchanged. We will then discuss the purpose of surfactants and how it manages surface tension within the alveoli. Within this discussion, we will also mention newborn respiratory distress syndrome and how surfactants play a role in premature babies. Finally, we will look at an interesting study that uses surfactants as a topical treatment for wounds. The respiratory system can be split into three parts: the upper respiratory system, the lower, and the alveoli. Let's work our way into the lungs, starting with the upper respiratory system, at our mouths and nasal passages where air and food enter the body. They converge at the pharynx, which determines if it is air coming in to send it to the trachea, or if it's food to send it to the esophagus using a small flap called the epiglottis. You can breathe through your mouth when you're sick because of the pharynx, since it is the passage for both the respiratory and digestive systems. The epiglottis makes sure that the correct passageway is open. When air enters, it first passes through the larynx or our voice box. When talking or singing, the glottis or laryngeal opening is open so we can speak using our vocal folds. Past the larynx is the trachea, which is a strong, muscular tube lined with mucus and cilia. This brings us to the lower respiratory system. The trachea branches out to the left and right bronchi, which enter into their respective lung. The lungs are surrounded by two membranes, and the visceral pleura membrane is the inner layer of the lung. The lungs are surrounded by two membranes, which I just mentioned. But between the membranes and the lung is a small space filled with fluid that reduces friction during expansion and compression. Each bronchus divides into many smaller, shorter bronchioles. At the end of the terminal bronchioles are alveoli, the final part of the respiratory system. This is where the exchange of oxygen and carbon dioxide come into play. The alveoli themselves are small sacs that look like grapes. They are made up of small layer of type 1 alveoli cells. The space between the blood capillaries and alveoli is minuscule, 50 times thinner than a sheet of paper. The total surface area of the capillaries to alveoli is around 807 square feet, about half the size of a volleyball court. Oxygen is used in cellular respiration, creating energy for the body. When we inhale, our lung volume expands and the pressure reduces. The partial pressure of carbon dioxide is greater in the capillaries, and the partial pressure of oxygen is higher in the alveoli. As we inhale, the carbon dioxide diffuses into the alveoli where there is lower concentration. Oxygen, in return, enters into the capillaries via diffusion and is attracted to hemoglobin. When we exhale, the carbon dioxide we just diffused over exits our lungs and our lung volume decreases, increasing the pressure in the cavity. Again, I must reiterate the importance of the lungs is the exchange of gases. The alveoli are critically important to this process. Inside the alveoli is a layer of water. Water is polar and is extremely attracted to itself because of the hydrogen bonds it can form. Therefore, there is surface tension in the alveoli, like you would see a bug skimming over a river or a lake. The tension keeps the layer of water stable, meaning it does not want to expand but stay in the same shape. Additionally, the water molecules would prefer to be as close as possible, making the expansion of alveolar cells difficult. With this knowledge, it is safe to assume that at some point the alveoli would collapse onto itself in an attempt to shrink the surface tension. We obviously don't want that. We want to be able to diffuse gaseous carbon dioxide out from the lungs without complete collapse so that oxygen can enter. Thankfully, type 2 alveolar cells in the alveoli create a substance called a surfactant. They are a mixture of proteins and lipids which form a foamy substance that decreases surface tension. Surfactants are dispersed through the alveoli, which decreases the amount of hydrogen bonds between water molecules. This allows the lungs to become more stable, decreasing the probability of lung collapse. Additionally, surfactants cause small alveoli more stability because there is less room for surface tension. Often we will see in premature babies that their lungs are not fully developed and do not have the ability to produce these surfactants. Therefore, every breath is the opening of alveoli due to great surface tension. An analogy for this is that these babies are trying to blow up a flaccid balloon with every breath. Within 2 hours, an artificial surfactant is administered. Babies are monitored for several hours or days, depending on the situation, and usually have good lung improvement. Surfactants are important to maintaining open alveoli for gaseous exchange and stabilization of the lungs. Aside from uses in the lungs, a 2020 study done in Germany researched whether or not surfactants could improve healing on external wounds. Wounds are characterized by inflammation, where epithelial cells resurface the wound and fibroblasts produce a new matrix. But inflammation is thought to cause chronic non-healing wounds like diabetic sores and scarring. The scientists hypothesized that surfactants may regulate macrophages and reduce inflammation. They believe this would reduce the need for plastic surgery for those non-healing wounds and scars. It would improve a patient's quality of life, reduce healthcare costs, and allow patients to work more frequently. The scientists needed a place to begin, so they gathered cell cultures from men and women. They were given permission to use alveofact, or ALV as I will refer to it, which is a lung surfactant that comes from cows. The scientists determined 2 concentrations, 0.01 milligrams per milliliter and 0.5 milligrams per milliliter, using the area of lung surface in newborns, which, as we know, can be administered surfactants. They began the first round of trials by scratching the cell cultures and placing 0.01 milligrams per milliliters onto the wound. The scientists saw that keratinocyte cells migrated quickly in the presence of ALV compared to the control. Satisfied with the in vivo experiment, they moved on to live subjects, mice. They scratched the mice and placed 0.01 milligrams per milliliters or 0.5 milligrams per milliliters on the wound. Macroscopically, there was no real difference between the control and ALV. But by day 14, the scientists saw that the mice with ALV had a thinner epidermal layer and less scarring, though the wound was longer. In mice with regular gauze, they had more scarring and a thicker epithelial healing. They also saw a reduction in inflammatory responses, including less cytokines that produced downstream of the inflammatory pathway. This means that less inflammatory molecules were needed. These 2 trials gave the scientists 2 positive responses. ALV did enhance the migration of cells and reduced pro-inflammatory responses. It was time to move on to human trials. Starting with 26 and trickling out several patients, the study was able to use 18 participants, both male and female. The participants were all wounded using a suction blister device. Each participant had both the control gauze on some blisters and ALV on other blisters, which gave the scientist great information to work with. They wanted to understand two things. Is using a surfactant safe and comfortable on a wound? And does it affect wound closure time? Using the standardized clinical scoring scale, they determined that ALV use on wounds were fine as compared to the control. As for wound closure time, that took more than a scale to measure. They collected data and created a percentage mean wound area on different days. They found that on days 2 and 4 that the results between the ALV treatment were healing faster than the control. Additionally, because of sex hormones, women had a smaller percentage mean wound area than men on day 4. But by day 6, both men and women had their wounds completely healed. This indicated that while ALV did not reduce wound healing time, it was more efficient. From the scientists' original hypothesis, they did see a regulation of migratory epithelial cells and a reduction of inflammatory responses. All ALV patients had less pro-inflammatory molecules like MMP3 and MMP9, both inflammatory enzymes, and IL-1 beta, a cytokine protein used in inflammation. There was also a reduction of tumor necrosis factor, or TNF, which is a response to inflammation downstream by the peripheral blood mononuclear cells. Like most studies, more information is needed and more trials will need to happen. This is a very promising start to see if surfactants can initiate wound repair processes or scar formation. The takeaway from today's lesson: lungs are the site of gaseous exchange. In the alveolar sacs, carbon dioxide concentrations in the blood move to the sac and is exhaled. During an inhale, oxygen is diffused from the higher concentrations in the alveolar sac into the bloodstream. The openness of the alveolar sac makes it easier to breathe and intake oxygen. Lung surfactants secreted by type 2 alveolar cells are a foamy substance that reduce the surface tension on the inner layer of water in the sacs. Surfactants are given to premature newborns to help with breathing. Additionally, surfactants could be used for wound healing in the future. In a clinical trial out of Germany, scientists tested whether surfactants would reduce inflammatory responses and migrate new cells. Mice trials showed a reduction of epithelial layers, causing less scarring. In human trials, they determined it was safe for topical use and showed a reduction in the inflammatory process. Surfactants, therefore, have many important uses. Thank you so much for watching my video today. This is a list of my resources, and almost all the images were provided by Google Creative Commons through PowerPoint. Thank you very much.